Hi everyone, thanks for joining me. Jake Robinson here. Thanks also to Susan for putting on a great conference, and also to the co-authors on this paper who are all listed on the front page. This talk is called Opportunities and Challenges for Microbiomics and Restoration Ecology. So microbiomics is the science of characterizing and quantifying molecules responsible for the structure, function and dynamics of a microbial community. It's generating new ways of thinking about ecosystems and their recovery. For instance, next-generation sequencing technology and meta-omics approaches can provide detailed information on how ecosystems function and how restoration interventions can enhance ecosystem health. And you can see from this first figure here, this is the first figure of our paper, and it just highlights some of the core functional roles that microbes play in our ecosystems. And so things like plant and animal health, nutrient cycling, and climate regulation. And this is the second figure of our paper, and this is linked to a historical review of microbiomics and restoration ecology over time. So you can see it starts thousands of years ago with the indigenous notions of restoring ecosystems, and then it goes to 1888 with Sergei Vinogradsky, who was the first person he pioneered the microbial ecology discipline. And then you go to the 50s and 60s, when DNA studies exploded and the genetic code was cracked. And then through to the 2000s when next generation sequencing came about, and it really fueled more microbiomics studies, and the advances in technologies allowed us to study microbial communities in the environment on a much deeper level. And this is the next figure in our study, and this really just highlights kind of opportunity zones or data equity issues across the world relating to microbiomics studies in restoration ecology. And so we looked at the SRA, so this is the Sequencing Read archive, and we looked at all the reads that were related to microbes in restoration ecology studies. And you can see some of the big players like North America, China, Australia, and then a few studies in Europe, and then some where there are potential data equity issues in places like South America, Africa, Russia, Indonesia, and Malaysia. And then this next study really is a sort of reconceptualization of ecological food webs. And so we have lots more knowledge now on how microbes interact with other organisms and energy flows in our ecosystems. So microbes play a role in predator-prey dynamics. Symbiotic microbiota can influence animal feeding decisions and sexual preferences. And so this really just provides an overview of a kind of more complex version of ecological food webs with considerations for microbes. The next figure in our study, from the top, this just shows you that the time from an ecosystem disturbance event through to ecosystem recovery, the ecosystem complexity increases across time. So we've studied things like species richness and community composition at the early stages, but then as species start to interact with each other, and then as communities start to interact with each other, the sort of ecological complexity increases across time. And we can use these advances in microbiomics technologies to understand these interactions, and then hopefully this will allow us to enhance restoration objectives. And so panel B in the middle just shows you how microbes and the technology associated with them can be used at each stage of a restoration ecology project, from the planning stage through to implementation and then to the monitoring stage. So you can use things like microbial inoculants in the implementation stage, and then we can sequence the environmental microbiome to see how the ecosystem recovers at the monitoring stage. And then the final panel at the bottom just shows you the different technologies and different pipelines that can be used to understand microbial communities. And things like compositional analysis and network analysis can be used in the early stages, and then through to functional information can be gathered from things like metagenomics, metatranscriptomics, metabolomics, and metaproteomics. And so this is just a review of how these technologies can be transferred to a restoration ecology context. Thank you very much for listening to my talk.